Myself, Dr. Mohit Kamthania. I'm faculty in Institute of Applied Medicine Research. I'm assistant for professor here. Today, my video lecture, my topic is splicing. So, in this lecture, we will include what is splicing and what is its significance and what is the mechanism of splicing. Simple. So, splicing means cutting. Simple. In case splicing simple include introns are removed and exons are joined to form the final mRNA molecule in case of splicing. So as per according to the central dogma of molecules from DNA, we just by the process of transcription there is formation of RNA or m-RNA, and from RNA by the process of translation there is formation of proteins. This process is very simple in case of prokaryotes, but in case of in prokaryotes, simple DNA, by the process of translation there is formation of RNA, or you can say mRNA, and then from translation there is formation of proteins. During after when the mRNA form, the process of translation continuously occurs. The ribosome comes and the process of translation immediately starts in case of prokaryotes. But in case of eukaryotes, there is some complex process and there is some variation, modification at the level of this mRNA. So in case of eukaryotes, there is different processing like RNA modifications, it includes there is 5' capping and 3' polyadenylation. And there is also some process like RNA editing. RNA editing, it includes editing of some nucleotides and removal of some nucleotides from the different region of mRNA, and it occurs with the help of gRNA, guide RNA. Okay, so and third is RNA splicing, which we are discussing this time. It includes removal of intron and joining of exons. So in splicing, when this in eukaryotes, this mRNA first form is known as pre-mRNA, and then splicing occurs, and then final pre-mRNA form. Splicing occurs, and then final mRNA form. Okay, so we'll discuss about splicing. There are different types of splicing, like self-splicing. This is intron-mediated splicing. In this case, introns have some signals, and it gives some signals, and self-splicing occurs in case of intron-mediated. And second one is spliceosome-mediated. Spliceosome-mediated splicing. There is spliceosome help in splicing, and spliceosome is a complex. It is a complex of SN, RNA and specific protein. SN, RNAs and some specific proteins. snRNAs and proteins, these form a complex which is known as spliceosomes. So, in splicing many spliceosomes help. So, we will discuss about this part. First, we know about what is spliceosome, snRNA, snRNPs. So, what are snRNA? snRNAs. These are small nuclear RNAs. Small nuclear RNAs. Less than 300 nucleotides long, and these help in splicing, and these form complex with some specific proteins, and that complex known as spliceosome. These RNAs are designated as U1, U2, U4, U5 and U6. These are snRNAs. U1, U2, U4, U4, 4, 5 and 6. These involve in splicing. These help in splicing. Okay. These specific proteins are this complex is known as RNPs. snRNAs. There's some specific proteins. It forms complex snRNPs, or it also known as SNRP. snRNPs means small nuclear, ribonucleoprotein particles. Small nucleoribonucleoprotein particles, these help in splicing mainly. So we will start with intron and exon. Suppose there is an exon, upstream exon, and there are sequences like exon, then there is adenine, then some pyrimidine, py means pyrimidine molecules, then N means any nucleotide, then cytosine. This is intron from GU to AG. This is intron, and there is downstream exon. For example, there are two exons, upstream exon, downstream exon, or intron. Okay, upstream it has 5. This is 5 prime. This is 3 prime. So it is downstream exon, and this is splice site. 5 prime splice site. Splicing occurs here, and this is 3 prime splice site. So splicing occurs there. This is intron. Actually, intron start with GU. Intron start with GU and ends at AG. Okay. In all cases, there will be start from GU and ends at AG. And there is in middle there is a branch site. This is a branch site. There is adenine molecule, adenosine molecule, residue, and these are pyrimidine, some molecules of pyrimidine. Pyrimidine means CTU, any molecules, not T as it is m-RNA, and N means any nucleotide, C that is in AG. So in this case, we have to remove this intron and join this exon first and exon second. So while in case of splicing, we do this. We will just write it in another way, like this exon 1, 5 prime, then GU start of intron. This is intron, a branch site, then end at AG. Okay, then next exon. Suppose it is exon 2, and this one is intron. This is branch site. So we have to remove this intron, GU to AG, and join this exon 1 and exon 2. So this process, first U1 and U2 add. So U1 and U2 are SNRNA molecule. These come and join at U1 join at GU, and it has some specific nucleotide sequence which attach with this area. So this is a consensus sequence. You will find this in the beginning of intron. This is a consensus sequence, and U1 has complementary sequences of these, and so U1 attach on this side. So this is exon 1, and U1 attach here by their complementary sequences, and then this is branch side. U2 attach on branch side. U2. And symbol this is exon 2. Okay. Now a complex of U4, U5 and U4, U5 and U6. This complex come and join in such a way that these exons come very close, so that the splicing process occurs, and these splice sites come near, and this introns will remove. Okay. So I am writing here. Exon 2, this is exon 1, and there are U4, U6, U4 and U5 complex. U6, U4, U5. It comes in such a way that GU or AG, and these exons come close, and then the branch side adenine molecule, this branch side adenine molecule, actually there occurs two trans-esterification reactions occurs, two trans-esterification reactions. These two prime OH group, hydroxyl group of second carbon, attack on this part, this phosphodiester bond. As this is a splice site, so second prime hydroxyl group attack on this phosphodiester bond, and it breaks phosphodiester bond and forms the bond with this. Okay, this G, and now it break this bond, and GOH as this is 5 prime, this is 3 prime OH group free. Now this 3 prime OH group attack on this part, this phosphodiester bond. So one phosphodiester bond formed here, and second phosphodiester bond formed here. So this first exon and this exon joined by the phosphodiester bond, and this intron will remove. So this is trans-esterification reaction. So two trans-esterification reaction occurs, as you see here, when this complex comes, it close it, and this adenine molecule 2OH second hydroxyl group attack on this, break this phosphoester bond, and this OH group, 3' OH group attack on this and form a phosphoester bond, and so these axons, axon 1 and axon 2 join as a splice product, or you can say final product or splice product, and this legate or intron removed from the complex. Legate, or you can say intron having these molecules of RNAs. So this is the simple process of splicing. There are almost 50 to 10,000 molecules in introns, and we will remove it and exons will join, and this is the final splice product. Thank you.